So what happens in lymphomas is that a double-strand break occurs, can be caused by a variety of things, and it's not properly repaired. So it's improper repair of a double-strand break. Some of these errors can be linked to mistakes or mutations in the proteins that are involved in the process. The problem is that a lot of the mutations are unidentified, or in eukaryotic model systems, they're lethal. So if you have a target protein you're interested in that's involved in recombination, you know it's important in recombination, but you make a mutation in a mouse, often those mutations end up being embryonic lethal and you have no mouse to study. Production of human mutations is essentially random. You cannot direct the mutants of interest, so you have to wait for someone to come into your facility and be sick, and then you have to try and figure out what's wrong. And the problem is it's such a very complicated process that we don't know what's wrong, and it takes a long time to figure out what's wrong because there's so many proteins involved and it's so very complicated in humans. And your mouse model gave you nothing because they died. So a lot of the work has been done in yeast, because a lot of mutations are not lethal in yeast. But the problem comes in that the proteins are very hard to handle from yeast. Being eukaryotic proteins, they're very, very fragile. So we've turned to the Sophilobus model because mutants are easy to get because they're essentially microbial organisms, so mutants are very easy to get. Proteins are easy to purify, they're easy to handle, and we can ask all the direct questions that you can't ask in a eukaryotic model because of the lethality problems. So to understand recombination as a process, we're trying to build recombination from the ground up using this model organism. They do everything that eukaryotes do with regard to DNA repair, they just do it much simpler. So we think that if we can reconstitute recombination in a tube using these archaeal proteins, that we can better understand how humans are doing it. And when it goes wrong in humans and causes a lymphoma, we'll have a better idea of what happened so that we might have a better target for treating lymphomas and other cancers.